Antigen and antibody interaction. In the previous videos, we discussed about what is antigen and what is antibody. We also learned about immune response. Antigen-antibody interaction is a kind of immune response. It is a response done by the acquired immunity. Acquired immunity shows two kinds of immune responses. One of the immune responses is humoral immunity or antibody-mediated immunity. Antibody-mediated immunity involves the production of antibody and the reaction between antigen and antibody. Antigen and antibody interaction involves three stages. In the first stage, there is formation of antigen-antibody complex. After the formation of antigen-antibody complex, reaction begins. The reactions are precipitation, agglutination, opsonisation, neutralization. Precipitation, agglutination are visible reactions. Due to the reactions, the pathogen will be destroyed. The last stage of antigen-antibody interaction is destruction of antigen or its neutralization. Some of the pathogens produce toxins. The toxins are neutralized by neutralization reaction. So there is binding force between antigen and antibody reaction. The binding forces are due to some factors. The closeness between antigen and antibody. When the antigen and antibody are closely fit, the binding force will be stronger. When the closeness between antigen and antibody decreases, then the force of attraction will be less. Antigen and antibody binding involves non-covalent bonds or intermolecular forces such as hydrogen bonds, electrostatic bonds, van der Waals forces and hydrophobic bonds. Affinity of antibody also influences the binding force of antigen-antibody reaction. Affinity of antibody, it is nothing but the attraction between the antigen and antibody, that is the attraction between epitope and paratope. We had already learned about epitope and paratope, and epitope is a part of antigen that binds with antibody. Paratope is a part of antibody that binds with antigen. It is the strength of the reaction between a single antigenic determinant and a single antigen binding site of the antibody. And a single antibody has two paratopes. But here the affinity of antibody defines the strength of reaction between a single epitope and single paratope. Antigen-antibody interactions is used in laboratory techniques. There are some applications. Let us see the applications of antigen-antibody interactions. These interactions are used in serological test of blood compatibility. You know what is serum. When the blood comes out of the body, it forms a clot, but the serum remains liquid. The serum has antibodies. This serological test, the serum antibodies are tested for blood group compatibility. During blood transfusion, the compatibility between A, B, O blood grouping is necessary. The antigen-antibody reactions is the basics for A, B, O blood group determination. And these reactions are used in the serological test of various pathogenic infections. When there is pathogenic infection, our body will produce antibodies. The antibody will be in the serum. When the serum is tested, we can understand whether pathogenic infection is there or not. And this antigen-antibody interaction is used in immunoassays. The diagnostic assays of AIDS, that is ELISA and Western blot. ELISA and Western blot are immunoassays. They are based on the antigen-antibody interactions. These interactions are used to detect the presence or absence of protein in the serum. Particular protein in the serum can be determined. And these interactions are used to determine certain immunodeficiency diseases. Immunodeficiency diseases are some of the immunodeficiency diseases are genetic in origin. Some of the immunodeficiency diseases are due to defective functioning of immune system. That can be determined by the antigen and antibody interactions. These are the applications of antigen and antibody interactions. There are different kinds of antigen and antibody interactions. The first one is precipitation reaction. And reaction between antigen and antibody results in the formation of precipitation of the pathogen. This reaction involves the reaction between soluble antigen and antibody. When the antibody reacts with soluble antigen, that causes precipitation. So visible precipitate is seen. In this reaction, the antibody is called as precipitin. Here antigens, here antibodies. The antigen antibodies react and form a precipitate. Another reaction is clumping or agglutination reaction. Here there is reaction between particulate antigen and antibody. Bacteria and red blood cells are the examples of particulate antigen, and the reaction between particulate antigen and antibody causes agglutination or clumping of the pathogen. The pathogens form a clump. When there is blood donation between mismatched blood group, that will result in agglutination of RBCs due to the antigen-antibody reaction. Opsonisation is another antigen-antibody reaction. This reaction involves IgG antibodies and IgE antibodies. IgG, IgE are types of antibodies we had already discussed. Here the antibody molecule does not destroy the pathogen. The antibody molecules form a coating around the pathogen. Opsonisation means coating over. Antibody molecules form a coating over the pathogen. Opsonisation involves the binding of an opsonin, that is antibody, to a receptor on the pathogen's cell membrane. Pathogen cell membrane has receptors or antigenic determinants. They are recognized by the antibody molecules, and the antibody molecules bind. The antibody molecules form a coating over. Pathogens are coated with opsonin. In this reaction, the antibody molecules are called as opsonins. This is antigen. The antibody molecules form a coating over the antigen so that the antigen is recognized by phagocytes. Phagocytes involved in phagocytosis. Phagocytosis means cell eating. Pathogen is marked out for destruction by the immune system. The coating over of antibody molecules on antigen acts as a marking. The pathogen is labeled so that other components of the immune system, especially the phagocytes, recognize the antigen. After the opsonin binds to the membrane, opsonin here is antibody. After the antibody forms a coating over with the pathogen, phagocytes are attracted to the pathogen. The attachment of phagocytes is facilitated. So this is enhanced attachment. The binding of antibody does not destroy the pathogen, but the binding of antibody over the surface of the pathogen facilitates the attachment of phagocytes to the pathogen. This results in much more efficient phagocytosis. The phagocytes engulf the pathogen and destroy the pathogen. Monocytes and neutrophils are the phagocytes. Already we discussed about phagocytosis while learning the innate immunity. Neutralization reaction involve the reaction between antibody and toxins. Some pathogens produce toxins. Here the antibody molecule acts as antitoxin. Elimination of harmful effect of bacterial exotoxins or a virus by specific antibodies come under the neutralization reaction. Here the antibodies act as antitoxins. The antitoxins neutralize the harmful effect of the bacterial exotoxins or viruses. This specific antibody is produced by host cell. Here the antibody is called as antitoxin. It is produced by the host cell, the infected cell. It is a response. The host cell produces antitoxin in response to a bacterial exotoxin or toxoid. Toxoid is inactivated toxin used in vaccines. After vaccination or after a bacterial infection, antitoxin is produced. But the toxoid introduction during vaccination produces antitoxin by the host. So it develops immunity. So toxoid is a kind of vaccine. In the next part, we will discuss about different kinds of vaccines.